Hey guys, welcome back to Sci. In this lesson, we're going to be continuing on protein synthesis and moving on to a step called translation. Now, if you remember from last video, we were talking about transcription. And if you want to check that video out, make sure to look at the last video in our playlist. So as a recap of transcription, you have your DNA, which is then transcripted into mRNA or messenger RNA, which is now moving out of the nucleus. So as it's moving out of the nucleus, it meets a ribosome. So we have a ribosome right here. And there's a lot of parts in this ribosome. As we talked about in our lesson on the endoplasmic reticulum, the nucleus, and ribosomes, our ribosome has two parts, and it's actually made up of RNA. So this RNA is made up in the nucleolus, and if you want to review that, make sure to check that video. But again, just as a recap, you have a large subunit and you have a small subunit. And these two subunits make up the ribosome. Now we also have three different steps in our overall look at translation. So we have initiation, elongation, and termination. But before we get into those three steps, let's talk about what actually happens to the mRNA. So if you look here, I've written out a word called a codon. And that codon is found in the mRNA. Now what a codon is it's three base pairs. So it's a group of these three base pairs, and they're called a codon because each of them actually code for a amino acid. Now if you look here, you also have a second part. And this second part is called tRNA, or transfer RNA. And what it does is it takes the anticodon, or the complementary base pairs for the codon in the mRNA, and it creates the anticodon, which then corresponds to the methionine. And so in this case, UAC is methionine. Now methionine is actually a very special amino acid. And this is because it's the start codon. So AUG in mRNA is the start codon, which codes for methionine. And so all amino acids, all of these polypeptide chains begin with your methionine codon, or begin with your methionine amino acid. Now let's take a look back at transcription. And we were talking about codons back then as well. We had UAA, UAG, and UGA, and these are your stop codons. And so once your ribosome reaches the stop codon at the end of your mRNA, it'll stop making your polypeptide. Now that we understand the interaction between your mRNA and your tRNA, let's talk about what occurs in the ribosome. So again, you have your initiation, so you have your methionine, and now these amino acids are linked together. And so you get a peptide bond between each of the amino acids as your mRNA passes through the ribosome. And so as that happens, you have elongation, so the amino acids are linked together, and then you reach that stop codon. And then the polypeptide is terminated. So that mRNA is not used anymore by that ribosome, and your ribosome will move on to another piece of mRNA. Now let's look closely at what happens in this. So your ribosome has three different stages. You have the E stage, the P stage, and the A stage. And these correspond to exit, polypeptide, and attachment. So your mRNA is attached here, and it exits through this side of your ribosome. And the P stage is probably the most important because this is where you have that tRNA that corresponds to your anticodon, and you will get each of these amino acids, which are again linked together by these peptide bonds, and finally creating your polypeptide. So you have these three parts or sections of your ribosome that correspond to these three different stages, and this continues again until you reach that stop codon. Now let's look at the bigger picture. Your single mRNA codes for a single polypeptide. But for maximum efficiency, so you don't need multiple different mRNAs, you actually have what is called a polyribosome, or multiple different ribosomes using the exact same piece of mRNA at the same time creating multiple of the same polypeptide. And so you don't need multiple of these mRNAs. Instead, you just use multiple ribosomes, and so the DNA is not accessed as much or is not tampered with as much, which also protects the DNA. And so again, you have a polyribosome, so you have many of these ribosomes that are acting on this mRNA strand, and you get multiple of these polypeptides. Now these polypeptides, and just moving forward beyond these steps, beyond just translation, these polypeptides are then combined together with all the other polypeptides, because a singular polypeptide is not usually a protein. Usually a protein is made up of multiple of these different polypeptides and polypeptides coming together. And we've talked about this in our protein video. But that is the final stage of your protein synthesis. It doesn't end at translation. It ends when your polypeptides are grouped together to create your final protein. Now, I'm not sure if I went over this, but this whole stage is called translation. And the reason for this is because you have your nucleic acid, your mRNA, which is translated into your amino acid. And your amino acid is a different language. It's the language of proteins. And this kind of concludes translation. You have your mRNA, which corresponds to tRNA. And this tRNA finds the amino acid somewhere in the cytoplasm. And based on that codon and anticodon, you'll have initiation and then elongation as these amino acids are added on. And finally termination, where you meet that stop codon. And just in conclusion, you have a polyribosome that acts on this mRNA and gets multiple of these different polypeptides. And so this concludes the first part of our protein synthesis, or the first part of our gene expression chapter. There's much more, and please join next video to learn more about gene expression. Bye.